Mila's touching the bark. Can you feel that, Mila? Yeah, that's the bark of the tree. Yeah. There we go. You got it. Mila's eyes have always been so telling. They light up when she's really interested in something or excited about something. She opens them so wide, I feel like I'm always looking into them. She's probably around close to 4 years old here. And I look at this picture and there's scrapes all down her face. My mom said, you know, she's just kind of a clumsy kid. And I just remember thinking, Mila's never been clumsy. She's incredibly coordinated.
Ready, steady. Whoa, good jumping, Mila.
Between when she was 5 years old and 6 years old, I started realizing just something is really wrong here.
Here, Mommy.
She's coming.
Mommy.
Okay.
Mommy is Mommy.
I started taking her to more and more doctors, and everyone was perplexed. It's like as if she developed and then stopped developing. For 2.5 years, I was looking for the answer and being told that there is no answer or that I'm crazy. Eventually, I felt so desperate that I said, I'm putting Mila in the car and bringing her to Colorado Children's Hospital ER because I give up. The genetic results came back and they said, it's Batten disease. My first reaction was, finally, I have an answer. But then it hit me that this was just probably one of the worst diagnoses I could have ever received. Children that have Batten disease basically have epilepsy, Parkinson's, blindness, dementia all together. No cure, no treatment. No child has ever lived. I looked at Mila and I thought, how is this possible that she is going to die in like 5 years?
My wife and I were sitting around the kitchen table, and she turned to me and said, hey, Tim, have you seen this? And she showed me this post on Facebook about a young family in Colorado that was looking for help. And they needed the expertise of someone who could do whole genome sequencing to help solve a genetic puzzle.
Dr. Yu called me and said, I saw your post. I work at Boston Children's Hospital, and I'm going to help you.
What's her typical sleep schedule?
She usually naps 1 hour during the day.
We found an unusual mutation that turned out to actually explain what was going on with Mila. We came up with the concept to try to design a small piece of synthetic DNA that acts as a little patch that covers up her mutation and essentially makes the cell ignore the mutation.
When Mila falls asleep at night, I put my hands around her head and just feel her energy and think, please just give me some reason to believe that maybe we could stop this. Now we were being told that we actually might have a treatment. Okay, the race is on.
This was a level of difficulty higher than any previous one that Boston Children's had done because we actually had to develop the drug and make it along the way. Drug development is typically a process where you throw 100 drugs down a pipe and one of them comes out the other end, and it might take 15 years to do it. That was not an option for Mila. This is a disease that takes no prisoners. It goes very fast. This was a Hail Mary, so to speak.
This is a situation in which they had designed a drug from scratch, but the drug has never been in animals, it's never been in people. We needed to design and deliver a toxicology study as fast as possible because her rate of decline was month by month. On top of that, we were going to have to work with FDA to ask them special permission to move forward quickly, which was an exception to their usual way of working. It was a tough sell, and we knew it.
Good morning, Mila. Mommy's gonna open your blinds. Good morning, Mila. Good morning.
I wrote a letter to the FDA, and it showed how much she had lost in terms of each of her abilities.
2, 3. And then we're gonna, there we go. We're gonna stretch.
And I wrote,
Yeah.
I think Mila has months left before it's going to be too late for her to receive this treatment.
It was very clear that we needed to do everything we could as fast as we could, even faster than we could, actually. I sat all these people in a room and I said, don't tell me that for this type of study, it takes 12 weeks. We're not gonna do standard. I need your best.
We were racing against time, and Mila was getting worse and worse. The FDA had to approve the drug, and that was the biggest step.
In January, we heard from the FDA that we had permission to proceed with the treatment.
The day that she received her first dose, I thought, people told me that it was impossible. They told me that it would take 10 years and that it would never be in time for Mila. And here I am in Boston Children's Hospital, and Mila is receiving an entirely new drug named after her. And I thought, oh my gosh, how is this possible?
We compressed usual toxicology from, I'd say, a solid year to a couple of months.
Hi, Mila. Hi, the big bear.
My porridge.
And if you think about her overall decline, if we had had extra months in there, how much more would she have lost?
This is a disease that normally, over the year that we've been treating Mila, she should have continued to very quickly decline, but we haven't seen her lose much of anything. She's gone from 30 seizures per day down to between 5 and 15, and they're much, much shorter than previously.
Before her treatment began, her hands and her arms had become very tightly locked, which is something that you see with Batten disease. Now she will open her own hands, and if I hold her elbow up a little bit, she will slowly open her fingers and feel something. These are seemingly minor things, but they're enormous in terms of her quality of life.
One of the existential questions that you always ask yourself as a person who's invested in clinical and basic science training is, how do we bring these two things together? With Mila, we were able to do that.
10 years ago, would we have thought that we would design a drug for one person? I don't think so. But now this has become real.
That sound, Mila? Do you hear the wind blowing? Yeah.
Every single life matters. Saving one person, one child, has an incredible ripple effect.
Splish splash.
We'll learn a lot from her that will help other people with many other diseases. You know, Mila is the first. This is the future of personalized medicine.
So, welcome everyone. Our last panel on balancing safety and innovation. And I'll introduce our panelists, and I have some prepared questions, but I encourage the speakers and everyone in the audience to also post questions in the Q&A. So we're joined this afternoon by Dr. PJ Brooks, who is the Deputy Director of the Office of Rare Disease Research in the National Center for Advancing Translational Sciences, or NCATS, within the NIH. He is interested in accelerating clinical trials in rare diseases by moving beyond the one disease at a time approaches, including gene editing and oligonucleotide therapies. Dr. Brooks coordinates the NIH Common Fund's Somatic Cell Genome Editing Program and the PSCGT program, and he chairs the Interdisciplinary Scientific Committee for the International Rare Disease Research Consortium. Dr. Barbara Koenig is a professor emerita of bioethics and medical anthropology at the University of California, San Francisco. Professor Koenig created and led the bioethics research program at the Mayo Clinic in Rochester, Minnesota, and was the founding executive director for the Center for Biomedical Ethics at Stanford University. She recently retired from her role as the director of the UCSF program in bioethics, but continues to serve as an embedded ethicist role in the Center for Maternal Fetal Precision Medicine at UCSF, for which we're eternally grateful. She works with our team on various projects to analyze new technologies such as gene therapy in the broader social and historical context to develop a robust understanding of the patient-family experience and to create flexible local governance of novel genomic technologies. Dr. Kevin Prohaska is the Associate Director and Bioethics Consultant in the Office of Clinical Policy at the US FDA, where he has served for nearly 20 years. There, he advises the Commissioner and other senior FDA officials on principles of good clinical practice and human subject protections, and he provides ethics consultations on clinical trials that raise complex ethical and regulatory issues. He assisted in the development of the Common Rule, which regulates the ethical treatment of research volunteers for all research supported or conducted by the federal government. And Dr. Fyodor Urnov is a professor of molecular and cell biology at the University of California, Berkeley, and the scientific director of technology and translation at the Innovative Genomics Institute. His research focuses on advancing genome editing technology and pushing the boundaries of how it can be applied to solve real-world problems. Excuse me. Dr. Urnov is a pioneer in the field of genome editing and has been able to take new gene editing therapies from the bench to the bedside. He also has a diverse background in academia, industry, and the nonprofit sector. Dr. Urnov's main focus is to accelerate the path of CRISPR-based therapies for rare genetic diseases from the lab to the clinic, and to develop new CRISPR-based technologies to understand and treat common diseases. And Julia Vitarello is founder and CEO of Mila's Miracle Foundation. As you know from the video, she worked alongside a team led by Dr. Tim Yu of Boston Children's Hospital to develop a first ever drug customized to just one person, her daughter Mila. Mila passed away in February 2021. Julia presents Mila's story at scientific meetings and conferences across the country. Driven by hope and a sense of obligation to help other families, Julia is dedicated to turning Mila's story into a new treatment path to personalized medicines for children with rare diseases. Thank you all of us for joining us today. Julia, my first question is for you. You have been such a pioneer in helping the medical community understand the impact of severe diseases on the family and considerations for N=1 therapies. Can you tell us a bit about the conversations around informed consent for first-time therapy and how you thought about the risk-benefit considerations? And what can we learn from your experience as we consider developing new therapies in utero?
Yeah, thank you, Tippy, so much for including me, by the way, in this conversation, because it's one that obviously means a lot to me. And it's just a really important conversation to be had right now. So thank you first for including me. I think many of you probably know Mila's story. And if you didn't see the video that was played right before this, without assuming that you all do, just in very, very brief, my daughter Mila was a very, very normal, typical, outgoing, coordinated, verbal child that was very healthy, that had no medical concerns. But around 3.5 years old, she started tripping and falling. She pulled items in really close, like toys and books. She was stuttering, and more kind of neurological symptoms came on over the course of 2 or 3 years. After about 100 different visits to doctors and therapists, I gave up because everyone told me that I was crazy, and that these symptoms would resolve by themselves. And they got worse and worse. I eventually brought her into the ER in Colorado Children's Hospital, which is near where I live. And she was diagnosed with a very rare variant of an already rare disease called Batten disease. And initially I was very happy that I was not crazy. And I felt like if there was a problem, I could somehow fix it. I knew nothing about genetics or about rare diseases. And after learning about Batten disease, about 5 minutes in, I realized that it was one of the worst diagnoses Mila could have been given. I started a foundation because I thought other parents had done that. And I started reading everything I could about rare disease and speaking with doctors and scientists. I started raising money and telling Mila's story. But Mila was missing a mutation and she needed two, and no lab could find the other one. And so my story crossed paths with Dr. Timothy Yu, as Tippy mentioned, at Boston Children's Hospital. And what I could say in an entire 30 minutes, I'll say in just 1 second, which is that a lot of work and time, not only by Dr. Yu, but by many other people, I'm sure many of you that are watching were part of this as well, of a pretty Herculean effort, which turned into, within just 1 year, a personalized antisense oligonucleotide, or ASO, that was tailored just to Mila, in the sense that there was no other child in the world that shared her mutation. This drug was called Millicin and was given to my daughter 3 years ago. And it showed enormous hope and promise in the first year. It surprised us all in the sense that it not only kind of stopped her very rapid decline at that point. She had lost her vision. She had lost her last words. She could barely stand, but she was still smiling and laughing and fighting hard. But after the first year, in the second year, it was harder to understand whether or not it was stopping her disease. And in the third year, we noticed that, her disease was taking over, and that she received Millicin just too late for her. She was 7, and that was very late for Batten, CLN7, which is her variant. After a 3-year fight, I like to say that Mila's spirit left her body, because that's what feels most comfortable for me. And it's been an incredibly difficult year for me, because that happened just only 7 or 8 months ago. And the reason why I'm here today, and why I continue to be a voice in the world of rare disease, and this is very exciting to be part of something that's even more cutting edge in terms of prenatal genetic therapies, is that, of course, I wonder, what if Mila received Millicin 3 months before? She didn't have any seizures 3 months prior to Millicin. What if she received Millicin 3 years before, where she was a outgoing little child that was had no symptoms, really? She was skiing, she was rock climbing, she was singing the ABCs and swimming. What if she had received that 3 years before? So this topic today really hits home for me. And I want to bring it around to Tippy's original question, just so you had some context to this. Is going into Millicin, no other child had ever received this drug. No human had ever received this. So Dr. Yu was very, very good at setting expectations correctly. Mila's cells were returning to, restoring health with Millicin in a lab, but that was the lab. And I knew where Mila was headed if she didn't receive Millicin. And I knew that she would lose every ability, possibly besides hearing. But other than that, she would lose every ability and she would die in the next few years. And I also knew that there were decades of work, because Dr. Yu explained all this, and I learned this over time talking to many people in the antisense oligonucleotide world, that there had been decades of work in animals, and this wasn't voodoo science. This was something very legit that was very promising, and had shown great promise with Spinraza for spinal muscular atrophy. And I knew what I was up against. And really, the way I started thinking about this was, what is the risk? The risk benefit analysis to me didn't make sense. Those words didn't mean anything to me. What meant something to me was, what is the risk of treating Mila versus the risk of not treating Mila? And that's the way I have and continue to speak about the paradigm, which is a really important one, because it is, the risk benefit analysis, however we talk about it, is the base of the house. It's the cement that you're pouring in the base that we build this house on. And how we approach this is really important. I think that I knew that it was a long shot in the sense that Mila was losing all of her abilities, and it was 7 years old, and she was the first person ever to receive a personalized ASO. But I knew that if we didn't give it a try, that she would lose all of her abilities. And something that people don't always really understand is with a child with a fatal disease, it is not just death that is initially when you hear that your child has a fatal disease, it's the death. But I will say that watching your child lose abilities is something that is beyond sadness. It's beyond pain. It is something that I have no doubt has cut many years off of my own life and all the lives of the people around me. It is hard to get through every day when you watch a child lose abilities. And so, all of this has to be considered when you're looking at the risk of treating Mila versus the risk of not treating Mila. And so it helped. It greatly helped a lot in the first year. And not only did it help in the first year, even though it wasn't in time for Mila, it greatly improved her quality of life. And so my decision, I didn't spend more than 1 second considering whether or not to do Millicent. It seemed obvious to me that this was a lot better than not trying at all. And, I think often of do no harm. I am not a scientist and I'm not a doctor, but I think of that and I understand why it exists. But I would say that do no harm can backfire if it's interpreted in an overly careful way. Because, and this is really important for me to say this, is that obviously we need to do things that are safe. We need to be very careful that a drug that's being delivered to the brain of a child that has a neurological condition is safe enough that it's not we don't think is gonna kill them on the spot or make their lives worse, of course. But we do need to be safe, that's a given, but we have to be very aggressive. If not, we will never make steps forward. And I hope that we can be both safe and aggressive at the same time. And, inactivity is much more harmful in my mind than very, in a calculated way, being both safe and aggressive. And so I hope that helps explain kind of the dynamic from a parent's point of view, having lived this, and sets the stage for these further conversations. And I think that it's very important that future families that go through this, especially those that we're talking about treatments in utero, are really explained that this is still new and we're learning a lot. And that there's a lot of hope and possible benefit, but we're also learning and there's going to be a lot of losses along the way as well. And that's okay, because we need to learn and we need to do something. Otherwise, there are millions of children like Mila dying. So I hope that sets the stage and answered your question, Tippy.
Yes, thank you so much, Julia. I really appreciate that. I'm going to turn over next to then PJ Brooks, sort of talking about rare diseases. PJ, it's been so wonderful to see all the work that you've been doing at the NIH and the FDA to sort of pave the road for these rare genetic diseases. Can you tell us a bit about your vision for the future and where early therapy falls into that vision? And are there specific considerations for prenatally diagnosed rare diseases that will be important for us to consider? Okay, I'll go to somebody else until we sort that out. Okay, Theodore, I have a question for you. You've been such a pioneer in developing and implementing genome editing approaches, particularly for in vivo applications. What is your perspective for the application of some of these therapies to the fetal environment?
Tippy, thank you for the invitation. It's an honor to be here and represent my institute, the Innovative Genomics Institute. First, Julia, I will admit, I bow before your courage. I spent 5 minutes watching Mila's movie, sobbing like my 5-year-old daughter would if she fell on the soccer field and got a major scrape. So I bow before your courage, and that courage and Mila and Tim inspire me as an editor and all of us to action. And what is that action based on? Well, thank you, Tippy, and thank you, Stefan, for a spectacular introduction to the progress that genomic therapies have been making since the first gene therapy started in 1989. It's a gene therapy was proposed in 1972. First gene therapy, 1989. Look at the progress we have made. Lenti, AAV approved, ASOs approved. I'm running out of memory for all the genome editing trials. And the central, I'm going to make 3 quick points as they relate to fetal genome editing, all sort of informed by the fact that I've had this good fortune of having worked on this for the past 21 years of my life, including Dr. Huang. So good to hear your voice. One of my first pre-INDs I filed when Dr. Huang, and I'm so glad you're still on duty at the agency, was well over a decade ago. So the first lesson is this. Every single first-in-human application of genome editing has had to have scientific, clinical, and regulatory innovation. So in other words, the notion that we kind of pre-build everything, that would be a generic approach to in utero genome editing, and then we go to the agency, and I'm not saying anyone is advocating for that, is not the way to go. You build a program that is a use case scenario. Dr. Huang spoke about this very eloquently. And you come to the agency with your data package and you tell them, this is what we'd like to do. And I think the voice of the community, and again, Julia, thank you for your courage and for what you're doing for the N of 1 community, would be enormous. And in that regard, let me really, again, emphasize how much Tim, you and the antisense community and Lorem have done with respect to all of this, and to what extent my field, genome editing, is really like a younger sibling to the ESOs. So 2, I really encourage everyone who is not a gene editor working on this to turn to us editors for solutions to problems that you have. We've made amazing progress in the past 13 years since we filed the first IND for genome editing in addressing an extraordinary range of preclinical programs in every step of the way. And all the early stage successes, 22 subjects edited by CRISPR Vertex for sickle thal, 6 subjects dosed by Intellia for TTR, 6 subjects dosed by Editas for LCA, and critically, an open IND for in vivo editing for HIV from Excision. That speaks to a growing body of a mature regulatory perspective that the agency has on our field. And as Dr. Huang or Dr. Lapteva or Dr. Prohaska will be the first one to tell us, everything is indication specific. But having said that, the agency, I think, has done an amazing job putting things together so that sponsors of investigational new drug applications for innovative approaches could come to them. And my third point, and with that I'll close, is to, there's a Russian saying that goes, a pessimist is a well-informed optimist. So I'm going to be a well-informed optimist. Every single SAE in my field for the past 30 years was not anticipated in preclinical studies. The severe adverse event for gamma retrovirus on the Hôpital Necker study for XKID was never before seen in mice. The cytokine release syndrome that Steph Group and Carl June saw with Emily Whitehead on CAR-T had never been seen before. Carl told me that when Emily was starting to die, they thought that she had an infection of some sort. They'd never seen this before. In my immediate field, which is genomic therapies for the hemoglobinopathies, where I'm very proud to be partnered with UCSF and UCLA to hold the only academic open IND for genome editing, there was just an SAE, an AML, an acute myelogenous leukemia, that was developed by a subject on a patient on a genomic therapy trial for gene therapy 5 years after the subject was dosed. This was not predicted. Why am I going through this? Objectively, the one way to determine how to safely deploy genomic therapies for antenatal therapy is to advance them to first in human. No amount of preclinical testing can convince us that something will be safe. That's just a fact of our field. And so as far as the preclinical framework for what you actually do, thank you very much, Dr. Parenteau, Dr. McKenzie, Dr. Sanders, and everyone who spoke about this, for really highlighting the fact that editing is in vivo. Whether it was the work by my sort of genome editing alma mater, Sangamo Therapeutics, for in vivo editing in the liver for the lysosomal storage diseases on hemophilia, or the magnificent recent work by Intellia on TTR. I note that they have now transitioned to a second open indication for a completely different disease. And then, of course, the wonderful new work from Editas and soon to be Excision. The bottom line is, I think our field overall is rapidly accelerating to dosing larger and larger cohorts of pediatric subjects. To wit, this will happen for the hemoglobinopathies. And in fact, David Williams at Children's Boston has dosed a 7-year-old boy with sickle. Intellia has guided that the agency has reviewed their data and has allowed them to open a pediatric cohort. And the same is true for LCA and Editas. And this is really to Julia Vittarello's point about when do you start? Well, so bottom line is, I'm really grateful to Tippy, and with that I'll close, for gathering us all on, and also to the agency, of course, to speak about this, because for me as an editor, the progression from dosing adult subjects, which is happening all out, to advancing this to pediatric cohorts, because that's when the degeneration begins, is happening all out, forms a logical arrow into an antenatal clinical trial for genome editing, because that's where, as long as the unmet medical need is there. So I'll go back silent again, except to say, again, I'm going to have to talk through the tears here. I work with a parent of a child with Rett, and closely. Rett is a monogenic neurodegenerative disease. And when I spoke with her about safety, she said to me, Fyodor, don't talk to me about safety. Some things are worse than death. And Julia, when I heard you speak about watching your child regress, I really choked up again. We have to be mindful about safety. One severe adverse event on a trial like this. And having said this, I really want to salute the agency, the clinicians, the scientists, for 30 years of mindful development in a risk balance informed way. And I just want to represent my field as being able to step up to the plate. Finally, I mentioned the IGI, which of course is here, UC Berkeley, UCSF, and I mentioned Carl June and Steph Group at Penn. I really want to salute UCSF for all the work that our sister university campus has done in advancing genomic medicine. I really think this is a fantastic opportunity for the nonprofit sector, for the academic sector, and the government sector. And so how wonderful to have PJ Brooks, who has been an extraordinary steward for a really visionary program by the federal government, to share the panel with. PJ, it's so good to see you, and congrats on the amazing progress you've made.
Well, thank you, Fyodor. I guess I'll go next then. And yeah, it's always tough to go after Julia. I think we all have that same feeling when we hear the story. But I think, one of the take-home messages that I've got from Julia's story at the beginning is that it's now possible to go from the identification of a disease-causing genetic mutation to a therapy, a rational potential therapy, fairly quickly. And you saw that with the ASO, but there are several approaches there. There are ASOs, there's gene therapy. In fact, when I met Julia first, they were thinking about gene therapy, and gene therapy could ultimately work for Batten. And then of course there's genome editing. But any of those could work. If you've got a monogenic disease, these are the approaches we have. And it's going to sound like a crazy thing to say, but in a sense, the disease almost doesn't matter to develop the therapy. You can do it based on the information about the genetic mutation, and the question becomes, how do you deliver to what cell type at what stage of development? And in principle, you could do that all the way in the prenatal period. So the programs that we've been focusing on, I put links to all of them because I don't have time to go through them all, at NIH, really all involve trying to make these genome target therapies into the therapeutic platforms that they really are and really are intended to be scientifically, and get away from doing these things one disease or one patient at a time, because it's really always doing the same thing. There are people doing what was done for Mila now with other genetic diseases. Tim Yu's doing it, other people are doing it. It's the same process. You design the oligo, you test it, you do the safety studies. And the way you do it's essentially the same. So the PAVE-GT and the Bespoke Gene Therapies Consortium are focused on how much of a platform we can make out of AAV gene therapy. The Bespoke Gene Therapy program in particular, which is organized by the FNIH, it's a public-private partnership, and you're going to be hearing a lot more about that in the near future when there's the official launch of it. A key part of that is the involvement of the FDA Center for Biologics and Peter Marks in the design of the Bespoke Gene Therapy Consortium, which I think is a wonderful thing. And I think genome editing is kind of the next step and potentially the most impactful, because particularly for the CRISPR-Cas series of genome editors, the way you direct them within the genome is you use guide RNAs, and guide RNAs are oligonucleotides. And all of the work that's been done subsequent to Mila And then Mila sort of pushed at the FDA to think about how we regulate oligonucleotides, I think will ultimately benefit the genome editing field. So I think all of these things can ultimately come together. And we do have a program, the somatic cell genome editing program, that I've been coordinating with many folks at NIH that has been focused on many things, including better animal models and also better delivery systems to get to all the cells and tissues you'd like to get to. And as we think about the next phase of that, which we've discussed publicly, one of the ideas we have is to be able to support clinical trials. And we would like to do it with the requirement that they're done, including more than one disease. So essentially to force people to do this as the platform that it truly is. And that's the way I think to get it to the most patients as quickly as possible. So I'll stop there.
Thank you, PJ. Yeah, it's really wonderful to see that platform approach and see, how can we be more efficient about getting these therapies to more patients. Barbara, I want to go to you next, and it's just been so incredible to have you and Julia as the embedded ethics program of our center. And I wanted to just ask you to speak about prenatal gene therapy and how does it present society with some unique ethical challenges that we need to consider as part of this discussion.
Well, thanks, Tippy, for a wonderful day. And Julia, thank you so much for sharing your daughter Mila's story. I think I'm exhausted. This has been phenomenal, but it's amazing. My first career before I decided to get a PhD was in pediatric nursing. That's what led me into my work in bioethics. And I would say that because of that, I'm deeply aware of the narrative of suffering and hope that accompanies all of these diseases and how complicated this is. We have on the one hand, the hope for the one dose and a cure, which is incredibly important. But on the other hand, we celebrate, sometimes we don't celebrate all of this, get all of the different views on the table. And I think today's program actually did. We've gotten a very full and comprehensive view. I also want to say that this narrative approach, thinking about the narratives of gene therapy or gene editing, sounds very different than the work that we heard about, which was basically trying to turn it into an equation, sort of thinking about the relationship. We'll just think about the risk-benefit ratio. We'll add risk-benefit for the mother, for the fetus, and then we'll somehow do a calculation and come up with an answer. I think that's not how these stories are playing out in the real world. And I think that's not going to benefit if the FDA needs to find a way, I think, of figuring out how to incorporate some of these narratives into the way they do their evaluation and how to get it into the iterative sense of what is important, what is not important. That's one of the things that we've been trying to do at UCSF, which is to actually have an approach which involves an embedded ethics presence. So we always have someone in the room who's worrying a little bit, being a worrier, and also asking questions when that's important. We presuppose a lot of things depending on what kind of words we use. Just making the decision to call this gene therapy rather than gene editing or a gene study or a gene experiment really pushes us in a particular direction of the narrative. Because if we're calling it therapy, we're also presupposing that it is going to be a benefit, even though we don't know. So that's complicated. We also, the word therapy, as I said, assumes benefit. But in a way, we also have emphasized today how the uncertainty continues. Fyodor just mentioned the cases of some cases of cancer that were totally unexpected. Off-targeted effects of the gene cannot be guaranteed. And I was interested that a couple people today have mentioned the sort of famous quote by Donald Rumsfeld, the idea of the unknown unknowns, or what you don't know, you don't know. I should say that this actually goes back to Aristotle as well, but it's something that we're not going to be able to get rid of. It's not going to be solved just by putting a series of declarative statements in an informed consent form. And making sure that the family understands that. It's much more complicated than that. And we have to really develop a way of sitting with and managing and being with this kind of uncertainty in the face of innovation. And oversight of innovative therapies is one of the hardest things. So I think at UCSF, we're trying to also innovate on that side, not just on the science, but also perhaps do some innovation about how we actually gather information about patient preferences and how we convey information to the FDA. So I'm going to end there. It looks like Tippy has gone silent. I'm not sure why. So I'm going to take the prerogative of being here at UCSF and say, Kevin, I'm going to turn the microphone over to you to talk about the FDA.
Okay, I'm happy to talk about the FDA. And first of all, thank you everyone for inviting me here to represent the FDA. Although what I'm saying is my own opinion. And also I'd like to say special thanks to Julia Trello, for sharing the story about Milo. The patient voice is critically important to the FDA, and we value that and consider it in a lot of our deliberations. And we encourage sponsors and clinical investigators to have that as part of the package in many ways, so that we understand what the needs are out there. That being said, a number of things were mentioned during this panel discussion I'd like to touch on very quickly. I think one of the statements that was made is that the risk benefit is the foundation for a lot of all of the discussions and the decisions that need to be made. And that's absolutely correct. Although it's not written that way in our regulations, if you look very carefully, they are all about determining what the risks and the potential benefits are to all of the research and trying to mitigate those risks wherever possible. But I also absolutely agree that it's unreasonable to think that the approach we should use, or the threshold we should use, is first do no harm. That may sound nice, but in reality, in clinical research, that really, in many ways, is not the right threshold for clinical research, especially in the environment of life-threatening conditions where there are no available treatments. In many ways, we do need to push the envelope and use some of these innovative therapies. And then finally, another thing that was briefly touched on is the use of platform trials. And I do think that that's actually a great idea. It's been used in a number of other different environments at the FDA, and we encourage that as well. And then the final point that I would make, and I think some of my colleagues here have also made this point, is very critical to reach out to the Responsible Review Division at the FDA and speak with them early about the plans for product development. They have a wealth of knowledge that they're willing to share with you, and it would help move things along more quickly if there is a better understanding right up front what's going to be required, and you'll find them be very, very helpful. Thank you.
Sorry, everyone, I'm back. I'm not sure what happened to my hardwire. We just have a couple minutes left. I did want to ask one question that was from a previous panel where we talked about, given that everything is the gene therapies are sequence specific, and some of the testing that we do in animal models tests a kind of a different species of sequences. What are some of the best ways that we can develop and test therapies for human applications?
So I can speak, Tippy, if you agree, I could speak briefly about genome editing, and others will be, and PJ, of course, I should really highlight PJ's leadership of the SCG for developing new tools to look at this. Folks, us developers of genomic therapies are in a perpetual race against two things. The first one is the next SAE, which, as Barbara correctly points out, from Aristotle to Don Rumsfeld, we know is the next unknown unknown. And as I mentioned, most of them are never seen in preclinical models. Nobody wants them, but the only way the field moves forward is learns about them and innovates to make things safer. The other thing is to really highlight the extent to which I mentioned working with Dr. Huang on a pre-IND 11 years ago, is the extent to which our field has really raced to develop new technologies to give us very strong evidence that what our therapies are doing. Certainly my world, which is genome editing, its advantages, it's a thing by its nature, based on Jennifer's and Emmanuel's amazing discovery, that targets the DNA. So as I look at what we had 11 years ago, I marvel at the range of assays that we now have to address the genome-wide specificity profile of action bioinformatically and wet lab across a broad range of cell types and tissues. And critically, and I think this is key and a remarkable recent development in the field led by Shengar Tsai at St. Jude and also Keith Jung, which is the development of rapid and robust assays to look at the personalized risk. In other words, we all have SNPs, right? We now have assays to take an experimental editor and look at the generic human genome, at a large panel of human cells and tissues, and also at the genome of the human being whom we're approaching with our experimental therapeutic. Furthermore, as I think Dr. Sanders described, we're rapidly developing new ways to do genome editing. For example, we're going from double-strand break-driven approaches to base editing-driven approaches. So long-winded way of me saying, I would not at this point, and I know that's a very strong thing to say for a genome editor who puts things into the clinic, I would not think of specificity as the gatekeeper for advancing a first-in-human to an antenatal therapy. We have a mature constellation of assays. Nobody can guarantee safety until we go to first-in-human. But right now, looking at how many trials are already dosing subjects in vivo at the reasonable safety record to date, and with eyes wide open and complete transparency, as Dr. Koenig put it out on the informed consent path, with the parents and a deep mindfulness for safety and risk, we have a way to de-risk a CRISPR something well put together to file an IND with the FDA for an antenatal therapy.
Yeah, and maybe Tippy, I'll just follow up on that. I think the question you raised is a good one. We do have some efforts going on with this within the somatic cell genome editing program to address that. How do you bridge that gap from studying effects on the human genome and in animal studies? Some other things that might be considered are there are certainly other approaches there, and I believe that as we go into SCGE2, there may be other approaches that we hope to support going forward, because I think that more generally, how do you bridge that gap from animals to humans is crucially important in all of these therapies, given the point that Fyodor made that I completely agree with, that we do surprisingly often see that some of the SAEs in human beings are ones that were not predicted in animals. And I think addressing that, agreeing with it, and in some of the programs we're working on, there's a focus on, given that that's true, what is the minimum number of animal safety studies that are needed to be sure to have before we go into the clinic and treat a human being? We want to be safe, but I think we want to figure out what is the minimal amount to get the trials up and running. I should also say that is my own personal opinion too. I want to give the appropriate disclaimer.
That sounds good. Julia, I want to give you kind of the last word in this panel. You've listened to a lot of these talks. You've been kind enough to talk to us. What is some advice that you would give for our antenatal gene therapy community moving forward?
Thank you, Tippy, for letting me kind of give a last word here. For me, I've been part of this rare disease world for the last 5 years, since Milo was diagnosed. And mostly because Milo's story kind of made it into the spotlight quite a bit. I've gotten the chance to get to know many, many different families and children across so many different diseases. And rare genetic disease in children, to me, feels like a global health crisis. And I'm always shocked that people in this industry don't use terms like this that may seem, I was kind of afraid to use these terms because they seemed exaggerated, and I'm a crazy parent that wants to fix the system, but in reality it is. And, when you look at an estimated, no one really knows the number, but 60 million, children with just fatal and life-threatening rare genetic diseases around the world, that's pretty serious. And, the numbers are up there with cancer, with HIV/AIDS, with COVID. So to me, this is a major crisis and involves children losing abilities, dying, having terrible lives while they're alive. And, I look at this, and then I also see on the science side that we have a toolbox, which was seemed almost empty when Milo was, diagnosed. There was gene therapy and there was a lot of stuff in the pipeline and in the works. But now we have a toolbox which is becoming more robust, and the science is getting there. And then I see these 60 million children, and then I see a toolbox that's kind of filling up, and we're learning a lot. And I think, what if Mila, like tiny little Mila in utero, received Millicin? These are genetic diseases. We don't know when they start. You guys know a lot better than I do, whether, at what point the genetic disease starts taking a toll on that child. But the reality is the dominoes probably start to fall possibly before birth. And so imagine Mila receiving Millicin in utero. She might never know what Batten disease is. We as a family and our community and our direct and indirect family and our friends may never know what Batten disease looks like, losing all of your abilities and dying. And how do you live with now a broken family, in my case, and a sibling that never receives any attention because I'm taking care of Mila all of the time up until recently, and all the pain on the other side of having to learn how to live in this world without my daughter. It is going to be risky. We have to learn things. This is new, but we have to start somewhere, and we have to take those risks and kind of accept that there are going to be losses along the way, and there might be many, but we have to do that because it's scary to imagine injecting Millicin into a tiny Mila in utero, but it is a lot scarier to imagine not trying and having tens of millions of children and their families go through the loss in months and years of losing every ability and being in pain and in and out of hospitals and dying. That's a lot scarier to me. So I just think we need to kind of always go back to looking at the risk of treating a child that has a fatal or life-threatening disease where there are no other treatments versus not even giving it a shot. Like, I think once again, we need to be safe and we need to be aggressive. Thank you so much for including me in this discussion. This actually is extremely exciting. I see hope, I see promise, and I hope that we can work, especially with the agency, which was really amazing with Millicin and really thought outside the box in a very, very new paradigm, was able to react very well and allow for Millicin to happen, which is allowing for, all of us being able to imagine developing, a drug for an individual, whether it be, a 7-year-old or in utero. So hopefully we can continue moving to be able to see more progress. So thank you for including me.
Thank you so much, Julia. And to all our panelists, I think that wraps up the day. I really want to thank the organizing committee, the speakers, and all the panelists for all your insights. And thank you to everyone in the audience for your attendance and engagement. You can definitely contact us with any questions in the future, and all of these posts are going to be posted online so that your colleagues can access later. I do hope, as Julia said, that this will serve as a concrete foundation as we continue to acquire knowledge and create dialogue around this important considerations for moving forward with a clinical trial. Thank you so much.